So my name is Muhammad Abbas, and I am PhD student under the studentship of DTP Food Biosystem, and I am working at University of Surrey. My second university is Queen's University Belfast. So I got this opportunity in 2022, and it was very hard. It was a big competition. So talking about the food biosystem, the incoming 2 decades, the food security will be a big challenge for us. So we need to prepare ourselves for that big challenge. So for the reason, if we are talking about the food and agriculture system in the UK, you can say it is contributing 6% in the UK economy, and its contribution is more than 100 billion pounds. So what I have selected is the fasciola hepatica disease, which is present in the different parts of the UK, and we are working on that disease. So if we talk about this disease particularly, it is costing 13 million to 40 million pounds to the UK economy annually. If we see globally, this disease is causing huge economic losses, more than 2.5 billion pounds. So we choose this project. And why I choose this project while applying in DTP? Because I belong to Pakistan, and this same disease is also prevalent there. So I can get benefit by myself for my country also. So in a broader perspective, this project is beneficial also with not only with the UK, it is with the internationally. So specifically with our project, our first aim is to develop the diagnostic system to diagnose this disease more specifically and with deeper sensitivity. So far we have developed a technique qPCR by which we can diagnose this disease in real-time manner, and this system is working well in our lab, and we have tested 430 samples so far from the different areas from the UK. From the cattle samples, we got more than 10% positivity, and from the sheep fecal samples, we got more than 50% of the positivity ratio with this disease in different regions. And previous literature shows that there is condemned or infected livers in the abattoirs up to 30%. So we can imagine the economic losses linked with this disease. And this is the first part of the project. And when we go to the second part, so far we have developed a whole genome sequence from this particular parasite, and we will use this whole genome data in the third part, while we are developing a panel of markers by which we can study the genome-wide structural changes with the DNA of this particular parasite in a single tube. So we have developed a panel of 12 markers, and we are going to apply that panel of marker on the field sample. And finally, we will update the forecasting model which was developed in 1950s, and still we are using it is valid, and we are using this model for forecasting in the coming years. And but what we want to develop is we need to incorporate our genetic data into this model so that we can see the genetic changes which may prevail in upcoming years in different geographical areas of the UK. So this is the overall summary of the project and what we have done so far and what we will do in upcoming 2 years. So this is what from my side for this project so far. Thank you.